So I don't see any comments, so I assume you can hear me and see also my slide.
Yes, everything looks and sounds good.
Okay, thank you very much. So I move on. So I guess in this audience, I can quickly go over this. As you know, coronaviruses are diverse. There's 4 genera known, and the alpha and beta coronaviruses have members that are human pathogenic. And this section of the corona phylogeny highlights the spectrum of diverse coronaviruses that can infect humans. So the novel zoonotic ones are highlighted in red, and they map to 2 different subgenera of the coronavirus phylogeny. And then there's in light blue, this more prevalent and common coronaviruses that map to another branch of the beta coronaviruses and a sub-branch of the alpha coronaviruses. So in a nutshell, given the spectrum of diversity and the recent emergence, it's likely that other coronaviruses diverse maybe around the corner. And so this merits also longer term efforts to screen and identify ultimately and develop antivirals that may be of use across a spectrum or maybe even a whole range of current and maybe future coronaviruses. And so we have started some maybe 10 months ago. We are not a coronavirus lab to begin with. And so we started this about 10 months ago with a couple of campaigns looking at complementary libraries that we received from collaboration partners, including the Scripps University here or the Scripps Research Institute from the US. And some of these are dedicated libraries. For instance, the Helmholtz library is heavily biased by natural products, which Helmholtz has been collecting for decades. Also BASF, a big German pharma company, actually not pharma, a chemical company, which is no longer in pharma business, nevertheless has huge compound libraries. They send us dedicated libraries towards specific targets that they pre-selected from 2 million compounds for being addressed towards specific enzyme targets of SARS-CoV. For the purpose of today's talk, I'd like to summarize how far we got in the area of repurposing compounds, which we received in collaboration from the Scripps Research Institute.
That's good.
Somebody has the mic. Okay, thank you. Now turned off. Thank you. And our approach is a little bit different from the common approach, and we did this for 2 reasons. First of all, technical, because we were not the first to start with the ReFRAME library, and so we wanted to distinguish ourselves from a previous campaign which directly started with SARS-CoV. We rather, on the opposite, started in our preliminary screening with an alpha coronavirus, 229E, and then in the rescreen switched to a German isolate collected in Bavaria to SARS-CoV in the counter screening. While this has certainly limitations, it on the opposite has the advantage of focusing on another branch of phylogeny of SARS first. And it also prioritizes non-entry inhibitors because these 2 viruses have different receptor usage. And so we were prioritizing molecules, maybe not directly on entry, because that's also complemented already by the antibody-based strategies very well, focusing on other targets. This is our approach. We start with a high throughput in a simple cell system with a dual luciferase assay. We can score cell viability with the firefly luciferase and score the virus replication with another luciferase in the virus itself. And then we do the counter screening in CALU and other human cell lines with SARS-2 and follow up in primary human air-liquid interface culture and differentiated primary human cells. A few words to the library itself, which I feel is unique and a special achievement of the Scripps Research Institute collecting it and the Bill and Melinda Gates Foundation funding this. It's, to my knowledge, the largest repurposing library that has been collected, and these 2 double rings show its composition. So the outer ring shows you the representation of molecules in this library and the given stage of development. So the vast majority has already seen in clinical trials in humans. And the inner ring shows you the etiology or the target, the disease context where these compounds are developed for. Some anti-infectives, but the majority cancer, CNS, respiratory system. So it's clear that these molecules are prioritized based on manipulating physiological processes in humans, addressing human targets, host targets. So we feel and hypothesized it's a powerful resource to identify potentially host targeting antivirals and then work with this information towards drug development in coronaviruses. So this is the first results of the primary screening of these roughly 13,000 compounds. In the dual luciferase assay, we used very stringent criteria of selecting candidates here and arrived at something more than 130 molecules, which reduce the replication of this virus below 10% and actually increase cell viability because obviously cytotoxic effects are rescued. And then we do a counter screening of all those in those response curves. We had this much of the compounds from the scripts so that we can do this. They send it out obviously in limited amounts, so there's not much freedom to operate, but that was important. And it was then interesting that already at this stage, we saw 12 molecules overlapping with the SARS screening of the ReVA paper that was recently published, including, for instance, remdesivir, that was obvious, but also apilimod and this protease inhibitor here. For instance, in addition, we picked up 5 different cyclin-dependent kinase inhibitors among the top 20 candidates, including dinaciclib, for instance, here. That's the rank number 2 of the screen. Number 1 was remdesivir, but others like flavopiridol and so on as well. Building up on this, we looked into the databases. What can we find about these preliminary hits and the targets that they addressed? And we thought that's of use simply to get a broad picture of what are the potential molecular targets addressed by these compounds, which merit further attention for in-depth molecular follow-up of potentially host targeting antivirals against an alpha coronavirus. In total, 59 target categories were identified. To the left, you can see, again, now among these 130 hits, to which stage of development these map to. And it was interesting to us to see that many of the hydroxytryptamine or dopamine receptor targeting molecules were picked up. We did not follow up here in detail yet, but also numerous candidates of this class were hit in this 229E screening, and none of them scored in the SARS-2. And that makes us speculate that they may be related to inhibition of entry of 229E, which is clearly mechanistically different. In addition, as I already pointed out, we saw a couple of high-ranking CDK inhibitors here dominating, and then maybe of interest also an anti-inflammatory compound, an NRF2 modulators we picked up, and ARH modulator here, Fortress. As I said, we did the counter screening with SARS-2, and we used, first of all, on the left, the very same cell line that we had used for the 229E screening, simply to rule out the target cell dependent effects, so that we reproduce this screening now in the same host cell line. And we had a little bit cell tox issue in this replica of the screening, much more than in the initial screening. Remdesivir came obviously as a strong hit. It's a qPCR-based readout. And then again, a couple of CDK inhibitors came here. And this is the rank of these preliminary candidates. And in parallel, we had the other replicate of the compounds that were sent to us for validation. In the CALU assay, here we look at qPCR data in the supernatant of infected CALU cells, and the crystal violet staining shows you that some of them are already toxic at this. It's a single dose of 5 micromolar in the screen here, or at least change the morphology of the cell culture, as you can see in the staining. Finally, we also could do an immunofluorescent staining and then an imaging-based automated quantification of infection to score also the hits in a direct antigen detection. And you see that some of these molecules mentioned earlier, like flavopiridol, Fortress, also BIAS, a CDK inhibitor, came back. And this is the imaging where you can see the antiviral activity, and the layer is not completely untouched, but cells are still surviving. I would like to give you a bird's eye overview of what we found in terms of overlap in these different screening approaches with the molecules that we received from scripts. So this Venn diagram shows you among the 134 compounds that scored in the 229E screen, they were all channeled in the validation with SARS-2. And then we have these two cellular systems, the HU87 based and the CALU based. And what is in this lens here, these 7 molecules are listed below. This is the overlap of the 2 reconfirmation screenings in the SARS assays, 2 different cell lines. And then you can see the rank positions of the molecules in these 3 assays, the 229E and the 2 independent reconfirmation. These are the compounds that we found. So we confirmed these 3 candidate cyclin-dependent kinase inhibitors. And then we were still interested also in this maybe most rigorous confirmation with Fortress and aryl hydrocarbon receptor antagonist or activator that we found. It's interesting to point out at this stage that throughout the year the last year, there was a very interesting proteomics analysis published in Cell highlighting that not only does SARS-2 activate cyclin-dependent kinase activity upon infection, there was also the bioinformatic suggestion that there would be a couple of cyclin-dependent kinase target sites within the SARS-CoV-2 proteins. And also here, dinaciclib in that dose activity response scored against SARS-CoV-2 in the assays of this other collaborating group here in Cell, which we found interesting and supports the notion that in principle, manipulation of cyclin-dependent kinases could be a tangible way of addressing this virus. Since in the meantime, this other complementary study was published already that started directly with SARS, it's worth it to look at the overlap between these 2 screenings to get a broad overview of the overlap between these 2 studies and the new findings that we added to this. So this is the left side with the 229E screening hits, where this cake here also shows you whether that has been screened before in other campaigns, whether it's scored as a hit or whether no information about this was already available in the ReFRAME database. These are the 12 overlapping hits between the REVA study published and our screening, which is presented here, and then the rank positions in these 2 different screens where a couple of molecules shine up in both assays, which supports the notion that in principle, these are molecules that could be candidates for broad spectrum antivirals. And last but not least, this is a bird's eye view on the performance and the hit rate of these compounds in the various assays that we did. The larger cake here shows whether that was already registered as a SARS inhibitor in the ReFRAME library, whether we could confirm it here and whether it overlaps with the REVA study here. And these, in this lens, these 4 are remdesivir, these 2 inhibitors that come together from the published REVA work and our work as well. Last but not least, we have established this pipeline of validating compounds in primary human lung tissue culture models. We get the cells or actually resections from lungs from the lung transplant unit of the Hanover Medical School, take them into culture, redifferentiate them, and then can infect them with SARS-2 and have established a qPCR-based readout system to analyze this. And this is now quite small. We tried to get as many of the candidates back from companies as possible to validate from an independent source. But there's limitations because not many companies can deliver these days. So we could not look at all candidates yet. But what is interesting to look at these, this list. So we, of course, we confirm remdesivir, which cell viability is scored with the CT value of the GAPDH message here. We see a little bit of an effect of apilimod, which has also been published in the ReVer study. And then it was only 2 doses that we could do so far. The CDK inhibitors, as expected, are difficult in that primary cells and raise certainly cytotoxicity. Candidates that we find interesting are these molecules Bardoxolone and Omaveloxolone, which are also modulating inflammatory processes, which we see some antiviral efficacy here in the cell culture fluid. It's a reduction of 1, where in this case, for instance, cytotoxicity is not kicking in yet in the assay. So that may be interesting to follow up, particularly since also for other reasons, not direct potential antiviral effects, there is a clinical study ongoing, a phase 2 in the US that's completed, recruited, and there's a phase 3 study actually planned for evaluating bardoxolone in SARS-CoV due to its anti-inflammatory and antioxidant properties, which may be protective in severe disease. With this, I'd like to conclude with my summary. So we went at this stage to the ReFRAME library from the alpha virus perspective, reconfirmed with the counter screening by SARS. Regarding the alpha coronavirus, we found roughly over 100 interesting candidates mapping to 59 different categories of molecules, and 7 we find in overlapping activity also against SARS in CALO3 cells. It's interesting to see and note that the CDKs play, at least in these cell culture models, roles in controlling replication. And depending on the cell line, this can be druggable, but that needs, of course, further attention, whether that's a pathway. And other than that, I think the bardoxolone group of molecules may be of interest for manipulating inflammatory process and also maybe having some antiviral efficacy. With this, I'd like to acknowledge Sibylle Heid, Alina Mattei, and Melina Winkler, who left their alternative projects or their starting projects for doing this work, and many of the collaborators and the funding sources for supporting this work. And thank you for your attention.
Thank you, Thomas. Do we have any questions from the attendees or the panelists? And if not, I would like to ask, how do the compounds in this library compare to the compounds in other libraries that have been screened? Are they entirely unique based on their availability in various clinical trials?
So I think it is the largest overarching library that I have heard of, and that also Scripps, tells me, and they publish this, that it is the largest collection, most comprehensive, and other sub-libraries have been screened, and that has been done in many places. And this library has been shared by Scripps to something like 12 or so different labs in different continents. And the first really nice study, I think, on this has been published by the Chandran lab now recently.
Okay, thank you. I think we have a question from Steve Goff. Steve?
Yeah, hi. So I was just curious about the CDK inhibitors. Do we know, which CDKs? Are they specific, or are these pan CDK inhibitors?
Yeah, to be honest, I'm not an expert on the CDK inhibitors, but reading the literature and what's in the database, I was not able to see a preference for a specific CDK. So some of them have a spectrum of CDK 4, 5, 9, for instance. It's also not so easy to say, that's only highly specific for the one and not the other. So I would not dare to comment on a specific preference. I think that would need a much more detailed workup and more knowledge in the background.
And I guess we don't know what the CDKs are doing. I mean, whether they're phosphorylating, POL2 or something else, who knows?
Absolutely, that's an interesting question. And so far it's a proteome survey that showed and suggested some direct phosphorylation sites in the viral proteins as well, and including the prediction of which viral proteins may be phosphorylated. But they did not reach as far as directly showing that indeed the CDK or which one specifically would phosphorylate that site. That's not known yet, to my